I'm going to start about how important the patient population is to the research that I'm doing. None of my research would be possible if we didn't have muscle biopsies, because every cell that I've touched has come out of a patient's arm and has been grown in a dish for some amount of time. So I want to do a little bit about talking how biopsies go from a biopsy in somebody's arm to the laboratory and how I use them. So as Larry mentioned before, we have some muscles that we try to focus on. So usually a bicep muscle, which is more affected in FSH, or a deltoid muscle, which is less affected. All of the cells I'm going to show you data from today is from a family, a control family, and an FSH patient, all bicep muscles. So this is the muscles that I tend to use the most in my experiments because they're highly expressing DUX4. So you have this muscle, the surgeon, let's say, takes it out of the arm, gives it to us, we can put it in a dish. And then once it's in a dish, we can grow it for a very long time. And as Larry said, we freeze them down and ship them out to different scientists around the world. So we have this repository, which actually has 56 families of cells. So that's either a patient and a family member, a first-degree family member who is unaffected by FSH. So we have all of these cell lines, 56 families, which gives us a lot of material to work with, and we can grow them in a dish pretty readily. And people have shown before, but I'm just going to show one more example. This is what the cells look like. This is a recent biopsy we did. So this is about 4 days in culture after the muscle biopsy. So this is what the cells look like. And then also, as Larry was saying, we can do a skin biopsy, and then we plate the skin in this different kind of media that's making happy factors and lots of stuff that makes the skin happy. And then what's exciting about the skin biopsies is that you can actually see the cells grow out of the skin, and those are fibroblasts. So here I'm showing you, where's my mouse? Do you see my mouse? There's my mouse. So this is a piece of skin that we plated on a dish, and you can see all of these cells around it have grown out of that skin. So those are the fibroblasts that are growing and dividing in the dish. So then we can harvest those and then using iPS technology, turn them back into a stem cell and then put them into muscle lineage and then use that for experiments as well. So that's kind of how we have from a biopsy into the lab and how we could distribute this to the entire population of scientists around the world that are using FSH models. So then I just want to give a little background about muscle regeneration, because that's one of the models I use in the lab is the xenograft model, and we use the whole point of the xenograft model is that muscles regenerate, and we take advantage of that process in our xenograft model. So I want to give a little information so we're all on the same page about how muscle regeneration happens in a very simple way. So when you have a muscle injury, which you can see by this lightning bolt, here's just a muscle fiber. And then these cells that sit on the outside of the muscle fiber called a satellite cell. When you have a muscle injury, these satellite cells get activated. And when they get activated, they cause a proliferation of these cells called myoblasts. And again, this is what these myoblasts look like in culture. They can grow and divide. And you can see here, these myoblasts have taken over the entire plate that I was looking at. So this is in culture in a dish again. And then as these myoblasts proliferate, if you change their media, they can differentiate into these fused myotubes. And then this is what this looks like in a dish. You can see when you compare the top picture to the bottom picture, you can see there's more long fiber looking cells. And if you zoom in on that, you can actually see, I don't have my pointer here, so I can't see where I'm pointing, but all of these little dots is a nucleus. You can see multinucleated cells in a dish. And I'm just going to show one example of when you have them in a dish for long enough, they'll actually contract, and they can contract off of the dish. So one day I was looking at my cells and this one was contracting, so I took a video on my phone. So here is just a video. It's a little subtle movement, but if you look at it, you can kind of see the muscle just go like this a little bit. That's the muscle contracting in a dish. So we can make functional muscle in a dish, and it's a subtle movement, but I hope you can see it contracting a little bit to get an idea of what we look at. So this is spontaneous. They just spontaneously contract in a dish, so I happened to see it, so I took a video and share it with all of you guys. So now that we know about muscle regeneration, how can we use this in a xenograft model to show how muscle regenerates and the differences between FSH muscle and control muscle? So in the Emerson lab, in collaboration with Katelyn Wagner, we created this cell xenograft model, and we use mice. So we have this immune deficient mouse line that we use for all of our experiments. And we have this immune deficient model because when we engraft human cells into the mice, if you have a mouse immune system, it'll reject that muscle. So then it won't be useful for any experiments. So our mice are immune deficient. And then we start out by ablating the mouse's stem cell population just in the legs, because then if you have the mouse's stem cell population, they'll regenerate mouse muscle. We want to regenerate human muscle in the mouse. So if we get rid of the mouse's stem cells, then we only can repopulate with humans, so then we can watch the regeneration of human muscle and not mouse muscle. So then in our mice, we cause a muscle injury and inject human myoblasts. So these are all cells I just showed you, all from patient muscle biopsies. And then we can have the human muscle regenerate in the leg of the mouse. And then we can measure human gene expression and also visualize human cells in the legs of these mice. So I'm just going to show you an example of how I visualize human cells in a mouse muscle. So this is just a false colored image of a mouse leg to show you the different muscles in the mouse leg. The one that I focus on is this tibialis anterior, or TA muscle. And what we can do is remove that muscle and take a cross section. And then I'm going to show you an image of what this looks like. So what I'm showing you here is the whole mouse tibialis anterior, taking a cross section, so you're looking right down that angle. And all of the green are actually human cells. So we put an antibody on this muscle to show where all of the human cells are localized to. So everything that's green here is showing where human muscle is. And these are just two examples. So there's an FSH cell line injected into the top and then a control injected into the bottom. And then we can also zoom in so you can kind of see this is a 10x magnification and then a 20x magnification. So you can zoom in and see what the cells are doing in that leg of the mouse and how they've regenerated. So we can ask if they're regenerating differently, if it's FSH versus control, and we can ask a lot of different questions about how these cells are regenerating in a mouse muscle. But they're all human cells, so it's really exciting. So that's one way that we can visualize how these xenografts are localizing in the mouse muscle. But I want to take a shift in how we're using this xenograft model to develop RNA therapeutics. So just very briefly, I'm going to touch upon RNA interference and how it's being used in therapeutic discovery. So ignore that whole left side of the screen. So I'm just going to very briefly say what RNA interference, or what we call RNAi, does. So you have these small little pieces of RNA, which is called an siRNA, and they recruit, they're specifically designed to target one gene in the genome. So then it goes in and it recruits these molecules to that gene and it degrades it so it never gets made into a protein. So what I'm doing is trying to use a DUX4 targeting siRNA, so it only goes in and destroys the DUX4 mRNA so you never get DUX4 protein, is my goal. So it's really exciting time to be in RNA therapeutics discovery, because just this summer, this company in Boston called Alnylam got an FDA-approved RNA therapeutic. So this is not targeting FSHD at all. So this is a liver disease. So this one drug has been approved to treat neuropathy in these patients. But there is proof of concept that we can get a drug approved by the FDA with RNA interference in this sort of same technology. So how am I using this technology, and what are the differences between this technology and what I'm doing? So this is also a liver target. So one big problem with the RNA therapeutics is delivery. So what we're working on is how do we get these molecules delivered to the muscle? So a lot of times it accumulates in the liver, which is what they want for this drug, but how do we get our molecules delivered to the muscle? And that's something I'm working on in collaboration with Dr. Anastasia Korova and John Watts here at the RNA Therapeutics Institute. So they've developed the technology that can target these siRNAs specifically to muscle. So in collaboration with them, we've developed these siRNAs that specifically target DUX4. And I'm going to, again, just show one little graph, only one. So I'm just going to show one graph showing several molecules that I've worked with them to design. And this is in vitro. So this is cells in a dish. I've added these compounds onto the cells to see if I can knock down DUX4 expression. And the way that we read that out is we see DUX4 activates lots of different genes, as Larry said earlier. So we could read how much expression of these other genes is, and that tells us how active DUX4 is. So just as an example, in the control, it's all normalized to control. So the control has no drug. You have expression of these targets. And then when I add all of the different drugs, that goes down. So that's what we wanted to see. So we see block of DUX4 activation in all of these different compounds. So now that we have in culture the evidence that we're knocking down DUX4, we can translate that into the in vivo xenograft model I told you about before. But before we do that, how do we target these siRNAs specifically to the muscle? So working with Anastasia and John, they've come up with this group. It's called a DCA lipid group. And if they add this on to the end of their siRNA, they see massive accumulation of this in muscle. So this is not mine specifically. They've done this with a different target that they're working on, but they've designed these molecules for me where they target DUX4 and they have this lipid group. So what they did was they saw accumulation in the muscle. So all of this is just a control on the top. So you can see there's no red. So if you have any red, that means that there is accumulation of this lipid group siRNA in the muscle. So they see great accumulation of this siRNA in the muscle. So now what I'm going to do next is take these lipid conjugated siRNAs and test them in my xenograft model. So, yes, so they did subcutaneous dosing, yeah. So they actually, in this experiment I just showed a picture from, they injected it into like the shoulder of the mouse, and this was a leg muscle where they saw the accumulation. So it's going systemically throughout the body of the mouse and eventually the human as well. So it's a lipid conjugated to the siRNA. So it's just delivered naked. All right, so now what I'm gonna do is take these lipid conjugated siRNAs and deliver them systemically to the mouse and hope that I can recapitulate what I saw in the dish where I knocked down DUX4 and see that hopefully happening in the mouse as well. So that's what I'm working on now. So hopefully have some of that next year if we do this again, and some updates on where I am. But that's all I've been working on. So I'd just like to thank, this is our lab here. This is Charlie in the middle. And Alec, you know, Jing is over here, and Dongsheng are also in the lab. Jing has done all of these animal experiments with me. And of course, I'd like to thank Funding, FSH Society, MDA, and the Wellstone for supporting me while I'm being a postdoc here at the Wellstone program. So I'd like to take any questions.